Everyone. In today's section, we shall look into the history and scope of fermentation technology. Historically, fermentation products were mainly food products, but in recent years, an increased interest has been observed in the production of bulk chemicals such as ethanol or any other solvents. Specialty chemicals like pharmaceuticals, industrial enzymes are also being produced along with biofuels and food additives. Fermentation processes are nowadays used in agriculture for production of single cell protein and also for the production of microbial pesticides. First, let us look into the definition of the fermentation. The word fermentation comes from the Latin word fermentare, which means to leaven, while others confirm that it comes from Latin verb fervere, which means to boil, and both the terminology are in use. The earliest use of the word fermentation mostly referred to natural metabolic processes in which a substance breaks down into simpler substances, usually due to metabolic activity of the microorganisms like yeast or bacteria. In biological point of view, fermentation is a way of extracting energy from molecules, and it is one of the common metabolic process to all living beings, be it a bacteria, an archaea, or an eukaryote. The fermentation provides ATP, that is the energy by the degradation of organic nutrients, usually under anaerobic condition. In animals, fermentation occurs within the gastrointestinal tract by the microorganisms that resist in our gut. From the biotechnological point of view, we are witnessing a revolution in biotechnology that has a far-reaching implication in different industries like the pharmaceuticals, food and feed, polymer, nanotechnology, oleochemicals, material science, etc., resulting in the betterment of the society. So you can see that biotechnology is not just restricted to genetic engineering or recombinant technology or even molecular biology. It is the field which has moved into various other expertise, such as you can see the examples as we have provided here. So you can see it in the nanotechnology, the chemical engineering, and even in the material science today. The present scenario defines biotechnology as any technique that uses living organisms or substances to make or modify a product to improve the plants and animals or to develop microorganisms for specific beneficial use. People applied fermentation to make products such as cheese, wine, meat, beer long before the biochemical process was understood. In today, we know that fermentation is a process used to produce specific product by living organisms. Examples of fermentation process includes the production of simpler products such as baker's yeast and alcohols, as well as complex products such as therapeutic proteins, antibiotics, enzymes, vaccines, and genetically engineered materials. Fermentation processes should be carefully and critically monitored with regards to the culture conditions and the time of harvest depending on the desired product. You can see here an pilot scale fermenter. So let us look at the background or the history of the fermentation technology. The Asian production of cheese, yogurt and bread from microorganisms and various alcoholic drinks such as the beer and wine were developed during 4000 to 7000 years ago when the process of fermentation was first discovered. Later, it was discovered that microorganisms like bacteria, yeast, or mold hydrolyze the sugars when they lack oxygen and are ultimately responsible for fermentation. This process results in the formation of products such as the food and drink. Consequently, fermentation was perhaps first explored by chance since in earlier times nobody knew how it worked. During the prehistoric era, some civilization considered fermentation to be a gift from their gods. Scientific evidence for fermentation was first described by Louis Pasteur in the late 1800s. He demonstrated a theory known as germ theory presented the survival of microorganisms and their effects on the process of fermentation. Thus, avoiding the speculation that fermentation was happening naturally. No, it was happening because of the presence of microorganisms, which was proved by Louis Pasteur. In the 1850s and 1860s, Pasteur became the first scientist to study fermentation when he demonstrated that fermentation was caused by living cells, that is yeast, thus giving a biggest blow to the abiogenesis theory. Buchner, a German chemist, received the Nobel Prize in 1907 for showing that enzymes in yeast cells causes fermentation. Arthur Hardens and Hans won the Nobel Prize in 1929 for their work who showed how enzymes cause fermentation. A British scientist named Weisman developed a fermenter for the first time for the production of acetone during First World War. Fermentation-based antibiotics production technology was established. The first large-scale fermenter, about 20-liter capacity, was used for the production of yeast in 1944. In 1934, use of Submerged culture technique was realized for penicillin production, essential aseptic condition with good aeration and agitation were probably the most important factors which led to the development of carefully designed and purpose-built fermentation vessel. In 1950s to 60s, steroid transformation by fungal spores was produced. In 1960, saw the commercialization production of amino acid, that is the monosodium glutamate, which is a well-known flavor enhancer. 1960s to 80s, mycotoxin treatment and reuse of waste from animals, plants, and domestics was demonstrated. In 1970s, microbiologically produced enzymes used in grain processing, sugar production, fruit juice clarification, detergent additives were produced. 1980s saw the revolution of genetic engineering techniques wherein insulin production was started. Paradoxically, the term industrial fermentation usually refers to either aerobic or anaerobic process, whereas fermentation in biochemical context describes a strictly anaerobic process which occurs if pyruvic acid does not enter the Krebs cycle. That is to tell, we have already studied in cellular metabolism that your glucose molecules will be broken down to 3 carbon pyruvic acid, and this pyruvic acid under aerobic condition has to enter the Krebs cycle for the production of other substrate, followed by production of ADP, NADP, H and FADH2. And all these in turns will enter into the electron transport chain. Why? Ultimately to produce ATP. That is the meaning of from the biochemical context. Industrial fermentation is a term used in chemical engineering describes the process operation that utilizes chemical changes induced by living organisms or enzymes in a particular bacteria, yeast, molds, or fungi which produces a specified product. So this is what is the definition of fermentation from the industrial point of view. And this is why I told it is paradox when we compare to industrial fermentation what happens within our body. Okay, here it can utilize both aerobic and anaerobic condition, but within our body we say that only under anaerobic condition the fermentation can proceed. So let us look into the design of industrial fermentation process. What are required? We require a pure culture of chosen organism in sufficient quantity and in correct physiological state, sterilized, carefully composed medium for growth of the organism, a seed fermenter, that is a mini model of production fermenter, to develop an inoculum, that is the starter culture, to initiate the process in the main fermenter, a production fermenter, the functional large model, and the equipment for drawing the culture medium in the steady state. Cell separation, collection of cell, pre-supernatant, product purification, effluent treatment. Now, each of these points will be explained in detail in the future classes, but for time being, students, kindly remember that from A to C constitute upstream and E constitute the downstream of the fermentation process. Now let us look into what do you mean by the upstream and downstream processing. These are the two important terminologies in fermentation technology. What do you mean by upstream processing? The upstream processing is the bioprocessing phase in which the cell lines are generated. That is, biomass is produced at appreciable scale. What do you mean by biomass? The biomass is the amount, the bulk of cells that are being produced. So, the biological materials are either obtained from an outside source or inoculated and grown in the culture. So, whatever the cells we are speaking, which is required, which is the starting material of your fermentation, can be obtained from the outside source or inoculated and grown in the culture. That is, you yourself can grow it in the laboratory and you can use it in the form of culture under controlled condition to manufacture certain types of products. So what do you mean by the downstream process? The downstream processing, on the other hand, is the phase that deals with the extraction and separation of the products from its biomass. It also involves testing, purifying, and packaging of the materials. Hopefully, these two terminology is understood. Once again, let me make it clearer in simple sentence. The upstream processing usually involves your biomass or cell lines or cultures of microorganism or plant or animal material. These are the initiators for your fermentation process to occur so that you will get the required product. After my product being formed, what you need to do is to segregate, to separate your product from the biomass culture. And this process is known as downstream processing. What you do here is you harvest your product, okay? You extract, you use the technique for separation. There are various techniques for separations which will be dealt in detail. And you test your compound, purify your compound. After testing and purifying the compound, you package your compound for the commercialization of your product. Coming to the fermenters and bioreactor, a fermenter is a setup to carry out the process of fermentation. That is, now whatever we told, the upstream, downstream, and also we told about the cultures, the products, all this requires what? An equipment, okay? And that equipment is your fermenter. And whatever the process which is occurring in this fermenter is known as fermentation. The fermenters vary from laboratory experimental models of 1 to 2 liter capacity to industrial models. So you saw both the models in the slide. I'll just go back. You can see this is a pilot scale, that is the laboratory scale, whereas you can see this large fermenters, okay, which are the main fermenters where large quantities of your desired products is being produced. So it may be 100 liters capacity, which refers to the volume of the main fermenting vessel. A bioreactor differs from the fermenter. In the former is used for the mass culture of plant and animal cells instead of microorganism. Okay? The chemical compounds synthesized by these culture cells, such as therapeutic agents, can be extracted easily from the cell biomass. That is, from your microorganism, you can easily separate your products when you compare to the fermenter, wherein you use the animal and the plant cells. The design, engineering and operational parameters of both fermenters and bioreactors are identical, with the involvement of microorganisms as elicitors. In some situations, the distinction between the two concepts is being gradually obliterated. That is, it is getting vanished. Today, we interchange the word fermenter and bioreactor. We don't segregate them and say it is fermenter and bioreactor. Fermenters or bioreactors are equipped with an aerator to supply oxygen in aerobic process, a stirrer to keep the concentration of the medium uniform, and a thermostat to regulate the temperature, a pH detector, and similar control devices. With this introduction, in the next classes, we shall go into the details about the fermentation technology